Hi, my name is Hailey Poster. I'm a junior biochemistry, molecular biology, and math double major here at Wittenberg University. I work with Dr. Kunal Chatterjee studying the effect of oxidative stress on yeast transfer RNA, or tRNA, production. During protein translation, tRNAs play the role of carrying amino acids to the growing polypeptide chain at the ribosome. Normally, functional yeast tRNAs are transcribed with extra nucleotide sequences on the 5' and 3' ends, represented in purple, that are processed off in the nucleus before they are exported to the mitochondria for intron splicing. The 5' extra sequence is called a leader sequence, and the 3' is called the Taylor sequence. Sometimes tRNAs are exported and introns are spliced before the 5' and 3' end extensions are removed. This results in these tRNAs being non-functional or aberrant because the extra nucleotides at the 3' end block amino acids from attaching. Non-functional tRNA is useless to the cell, and under healthy conditions, there is only a small amount produced. There is little research exploring the concentration of aberrant tRNA under stress conditions. We decided to investigate the levels of yeast aberrant tRNA quantity under oxidative stress. Oxidative stress is produced when reactive oxygen species, or ROS, overpower the natural antioxidant defense system in the cell. ROS that are generated by hydrogen peroxide are destructive to lipids, proteins, and DNA, which contributes to tumorigenesis and neurodegenerative disease. Hydrogen peroxide stress has been shown to decrease yeast viability and increase tRNA fragment production. I hypothesized that under oxidative stress conditions, the amount of aberrant tRNA in yeast cells would increase. I first conducted a growth curve by growing the yeast in liquid YEPD media overnight until they reached an OD 600 of approximately 0.3. I then applied hydrogen peroxide at concentrations of 0.5 millimolar and 2 millimolar, as well as kept a control of 0 millimolar, and grew for another 2 hours before isolating the cells. The growth curve obtained diverges after the stress was applied. After 24 hours, the 0.5 millimolar cells were able to recuperate, but the 2.0 millimolar cells did not. However, the stress wasn't too much as to stop growth completely. After the cells were spun down, the RNA was isolated using the hot phenol technique, and the RNA was DNase treated. Then, reverse transcription was used to target the specific 5' and 3' end extended tRNA isoleucine and simultaneously target tRNA glutamine to act as an internal control. The forward primer for the aberrant tRNA bound at the 5' extension, while the reverse primer bound at the anticodon loop, where the introns were previously located, to ensure only tRNAs with end extensions were targeted. For the glutamine control, the primers bound at the 5' end with extensions already processed off and the anticodon loop, respectively. The samples were then PCR'd for amplification and ran in a 3% agarose gel. A 25 base pair ladder was ran alongside the samples. A -RT sample was also ran that lacked reverse transcriptase to ensure our tRNAs were the only thing being showcased. The first band represents our tRNA glutamine control, while the second band is the aberrant tRNA. Normalized ratios between the three bands were calculated using ImageJ software. The ratios and band intensity show that as hydrogen peroxide stress increases, the amount of aberrant tRNA decreases. This finding was consistent across my 3 trials and contradicted my hypothesis. I questioned whether this meant that transcription of new tRNA was hindered by the oxidative stress. I used the same RT-PCR technique, but this time used an aberrant tRNA forward primer that bound to intron-containing pre-tRNA isoleucine to detect if pre-tRNAs were still being transcribed. The presence of these introns in the pre-tRNA meant that the tRNA was newly transcribed and has not been out of the nucleus yet. I ran another 3% agarose gel. This time, the intron-containing aberrant tRNA band was on top because of the increased size in base pairs, and the tRNA glutamine was below. The normalized ratios and band intensities showed that under the 2 millimolar hydrogen peroxide stress, transcription on new tRNAs was hindered. However, the control and 0.5 millimolar hydrogen peroxide conditions resulted in nearly identical bands. Thus, new tRNAs are still produced at low-dose hydrogen peroxide stress. There can be various explanations to my observations. First, under oxidative stress, the levels of tRNA repair enzymes like RRP6 may be upregulated. Second, the level of tRNA exporters like LOS1, MEC67, MTR2, and CREM1 may also be mislocalized. This will give the cells more time to process the leader and Taylor sequences off before they are exported. In the future, we are exploring whether the levels of tRNA repair enzymes like RRP6 delta work in cells under 0.5 millimolar stress. I also want to investigate the other 10 isodecoder aberrant tRNAs that also have the 5' and 3' extensions and how they react under hydrogen peroxide stress to see whether my observation holds true for all intron-containing tRNA families. Lastly, I want to apply other stressors such as salt, caffeine, and nutrient-deprived media to yeast and observe how aberrant tRNA concentration is affected.